is Miss Smith, and I wanted to intro the sales cycle with you this week. So what we're going to basically cover is a quick intro to what is the sales cycle, why it's needed, and then I will cover the remainder of these slides later in the week. This is loaded in your Google Classroom, so you should have access to this as we go through. So first off, you need to understand that sales are tiny for a couple different reasons. First off, they have a certain job to do. Sales make tissue. So if sales are too large, they can't do the job they're designed to do. They're also there because they want to be there for protection. If you have something huge that receives a lot of damage, it's a lot of repair that's needed to that larger subject. So sales stay small so they can withstand that trauma that they may experience and that they can repair quickly versus something larger having to repair. Also, for efficiency of movement, that's what this is stating, surface area to volume ratio. If you have a lot of little things, the movement between one particle to the next happens more efficiently than if you have one huge, think of the bouncy balls at Walmart. If you have one big bouncy ball at Walmart, and somebody comes through and punches your big bouncy ball at Walmart, you're done. But if you have a lot of those little gumball machine balls and you lose one or two here and there, not so much a big deal. So when something is smaller, it tends to be more efficient because materials can move a little more effectively through the cell. There's a little bit of math here that I'm going to get into once we return face-to-face, which for those of you who are wondering, that is coming up soon. I think you'll get these dates soon, but October 5th is the return date for freshmen and sophomores, and the 19th is the return date for juniors and seniors, and that will be on an A-day, B-day schedule. More info will come. Once we get in person, I'll go over this math, which is fairly self-explanatory. It talks about how, if you're looking at a cube, how your surface area to volume ratio is much more effective if your cube is smaller. So this circle in the middle would be similar to a nucleus, and as you look at the smaller cube, you can move materials into the cell, into the nucleus a lot more efficiently. So if I'm looking at surface area of a cube, I'm going to take my length and my width and multiply it by 6. My volume would be my length, my width times height. So that would be a 6-to-1 ratio down here when it talks about surface area to volume. You want that number so that the movement of molecules can be greater. You want that 6-to-1 difference versus something that, or smaller numbers there, I'm sorry. We'll look at that again once we get in class in person. All right, so cell maintenance, why do we need to look at the movement of these cells? One is for the maintenance of cells so that they can repair when they need to, so that DNA can be produced when needed, so that we can move materials through the cell. We need to get water in and out. We need to get oxygen in and out. One of the main things that fuels your cells is oxygen. That's what goes into the mitochondria, the aerobic mitochondria, to make sure that cell respiration can take place. So we need small cells. We need to make sure we have enough DNA and surface area to volume ratio, which brings us to the cell cycle. And I'm going to skip a couple of these slides here and there, so please stay with me. First off, I don't like this definition for sexual and asexual, but there's reproduction that happens cellularly. We are cellularly asexual reproducers, which means one cell makes two. Two cells make four, four makes eight, eight makes 16, and they're identical to the parent cell. Sexual reproduction means there's variation in the offspring. Two individuals come together and produce the offspring, and there's that genetic combination there. But as far as cells go, we are asexual, meaning our cells, if a skin cell reproduces, it makes exactly copies of that new skin cell. You don't want a skin cell making anything else if it's going to be a skin cell. So that's important to realize. All right, I'm going to skip this. This is how we look at bacteria dividing or multiplying. So we're going to skip that. And this number becomes pretty important. We look at our mitotic division. Mitosis is cellular division. And when you look at that, you're going to be looking at a diploid cell, which would be like a liver cell or a cat's liver cell, a dog's liver cell. It doesn't matter. Whoever's liver cell we're looking at, except for sperm and egg. It says that here because those are sexual gametes or sexual reproducing cells, so they need less chromosome numbers. But and we'll talk about that later. But if we're looking at somatic cells, which are your body cells, your skin, your liver, your heart, your brain, your muscles, your nervous system, and so forth, we're going to look at that number being diploid in those cells. So you have a diploid number, which is 2n, of being 46. This is a human cell. You have 46 chromosomes. After that one liver cell undergoes cell division, it's still going to have 46 chromosomes. They're identical. Why does that happen? Because in interphase, I misspoke there. In interphase, the DNA replicates. So you have more DNA for a brief period of time so that this can happen. Science is the only topic where in division, your number doesn't decrease. It stays the same. The cell divides, but the chromosome number stays the same. Okay, so the cell cycle is basically divided into two main categories. Interphase, which is when your cell's normally doing its stuff. It's growing, it's making protein, it's adapting to its surroundings. And then you have mitosis, which is where you actually divide. And cytokinesis is kind of like a phase at the end. So when we look at these, they're divided into these categories over to the right. You have G1, S, G2, which is going to come up in a picture in a second. And then you have prophase, metaphase, anaphase, and telophase. Okay? So these first three are all a part of interphase, the G1, S, and G2. And again, they're in a diagram in a second. These last four are all a part of mitosis. Interphase is known as cell growth. Mitosis is known as cell division. So you have growth and division. So what is cytokinesis? That's just like a stage at the end where the cytoplasm, the jelly-like stuff on the inside, actually divides. Okay, so most of the time, you see this heading over here, interphase, most of the time your cell is going to be spent in its interphase. It's going to be growing. Why? Because that's when it grows, that's when it makes proteins, that's when everything metabolic that happens in your body is occurring, all the chemical reactions, and that's when your DNA replicates. All those activities take place in interphase. So the majority of your cell's life will take place during that time. So there's a diagram that helps us. This big, if this were a pie chart, you see that most of this pie chart is made of interphase. All right, look where I'm moving my cursor over here. Most of this pie chart is made of interphase. G1, which you do need to know what happens there. This is when normal activities are occurring, we're growing and so forth. S, that's when we're replicating our DNA. So like I said earlier, there's a point where DNA replicates. It is right here. And then G2, that's when we're getting ready to divide. Guess what's missing right here in this little piece of pie? Mitosis. Mitosis is the cellular division portion. It gives you a little information about interphase. I'm going to skip through these sections because this we will map out once we return as we do the map of a chromosome. Skip to this one. Okay, so right after interphase, 90% of that cell's life is growing. You roll into prophase. And prophase is going to be a stage where you're getting ready to divide, but it's actually part of mitosis. So if you remember pro and prep, prophase, you're prepping the cell by all the organelles basically getting out of the way. You don't see a mitochondria in here. You don't see a Golgi, but you do see a nucleus, and you see something unique to animal cells here. You see centrioles. So the nucleus is here, and there's chromosomes inside, and then you see that everything else is gone because we are prepping to divide. All right, so a little bit of vocab here. These are centrioles. These are spindle fibers, and they basically are going to attach to your chromosome and be pulled in one direction or the other. The chromosomes will. You can click these interactives as well. I'm not going to do that now, but you can click those interactives and get a more animated visual at what's happening here. Here's pictures of prophase. You can start seeing that some of the DNA looks squiggly. This is not prophase. But these are all pictures of prophase, different stages there. So early prophase, you start seeing the nucleus disappearing. These are centrioles. These are spindle fibers. Late prophase, you actually start seeing your chromosomes become visible. The spindle fibers are what attaches the spindle fibers to the centromeres. That's going to be kinetochores. These little, I call them lassos, but they basically are the part of the lasso that a cowboy will throw that connect to the centromere and pull the chromosome apart. Metamiddle. Metaphase is the middle where chromosomes are lining up in the middle. Notice the centrioles have already migrated. The spindle fibers have attached to the chromosomes, so we still have all that going. And we have this equator that has formed here in the middle of the cell. And what we're doing here is we're actually having the attachment, or the organization, I should say, of these chromosomes into that equator of the cell, the middle of the cell. It could also be lined up left to right here horizontally, and your centrioles could be top to bottom. It doesn't matter. You're just looking at this metaphase process taking place. Here's what metaphase looks like. It's pretty distinguishable because those chromosomes are in the middle. During anaphase, you're going to separate. So I usually tell students ana away, anaphase away or apart. You're going to have the separation of those chromosomes. And you see that here where the spindle fibers are pulling the chromosomes to each side. When I say each side, I'm referencing the centromeres here. And then anaphase here, you see that. And then you start really seeing that here. All right, and then telophase, I want you to remember tele2. You start seeing two, as it says right here, identical cells formed, producing the new cells that are going to form. The original cells no longer existing. These are called daughter cells. They are exact copies of the parent. So the chromosomes are going to start organizing into chromatin, which is basically squigglies. Your nucleus is going to start reappearing on both sides here, and your centrioles are going to start kind of forming and organizing. Your spindles are no longer there anymore. Telophase looks like this. You can see the division there. By the way, this is a plant cell because I can see the cell wall around the plant cell. And this is what it looks like as the cytoplasm is starting to divide. So you have cytokinesis developing here, and it kind of looks like a peach or another image. But anyway, you start seeing the development of two distinct nuclei. And then cytokinesis is where we actually get the cytoplasm dividing. So the chromosomes have already divided. We're just looking at the cytoplasm dividing now. And this is what things look like. This is prophase, where the chromosomes are visible. This is metaphase, we're in the middle. This is anaphase. The purple things are the chromosomes. The chromosomes are starting to pull away. And this is telophase because I can see two distinct cells forming. So I've got prophase, metaphase, anaphase, telophase, PMAT. Prophase, metaphase, anaphase, telophase. All right, and then a couple questions, and then I'll end with you here. If a plant cell undergoes mitosis, but no cell plate is formed, what would be the result? The result of that would be an overloaded cell. The cell would have too many chromosomes. The cell plate is what divides that. It says corn cells have 20 chromosomes. If it is divided mitotically, how many chromosomes will be present at prophase, meta, and ana? That's still 20. 20, 20, 20. Nothing changes. A biologist studies corn roots with 20 chromosomes, and he locates one in late telophase with only 19 chromosomes. That might have occurred because some of the chromosomes didn't separate properly if they only have 19. So 19 chromosomes could be a result of faulty mitosis, which happens, and we'll talk about that as well. All right, so I'm going to stop there, and I will include a Google form with a couple questions on it. The Google form will not be loaded right away, but will be loaded later today, so don't get too excited looking for that. And we will pick up with the remainder of these questions later on. So basically, I'm stopping with this slide with the questions here. So I hope you have a great day.